Thank you, David, and thank you, members. It's such a great honor to be here. And as I was listening to the introductions, I was thinking how special these meetings are. And the main reason is that surgery is unique in one regard. We do not learn how to be surgeons from a book or an app or a website. You know, we learn how to be surgeons by being taught directly from other surgeons, hand to hand, throughout our entire professional career. And no matter how long we've been in the field, we still need to learn. And it's at meetings like this where people get together, tell their war stories, talk about what's going on in their surgical careers, that we really learn and advance as a profession. So it's just such a great honor to be able to come here, participate in this meeting, and talk to you today. And it's also really special to think about an opportunity to describe a little bit about what Dr. Letton was able to accomplish in his career and how that fits into the overall fabric of what we're doing in clinical cancer research. So I tried to do a little bit of that with this talk. So I'm going to talk a little bit about the history of cancer clinical trials, how we got where we are today, and actually where I think we are today. And then also focus a little bit at the end of my talk about what surgeon-focused contributions can be, in particular, the kind of things to cancer clinical research that only surgeons can do. So all of us as cancer surgeons take as our first guidance the work of Halsted, more than 140 years ago now almost. When the disease was terrible, it presented most of the time in an advanced state, and really only the skill of the surgeon was able to save a patient. And remarkable back in Halsted's time, given everything, all the challenges of treating disease surgically, 20% of women who presented to the Halsted teams with breast cancer were cured of their disease. Truly remarkable for the time, and established him and his legacy as the standard for all of us, thinking about excellence with the tools that we have at hand. So next, I think things that are notable, worth thinking about in terms of clinical research too. 1907, the American Association for Cancer Research was founded. It was founded by 6 surgeons and 5 pathologists. And the surgeons, they're listed here on this slide. Because remember, the fields of medical oncology, biostatistics, many of the things that are involved in cancer treatment today were not present in 1907. In 1914, the American Cancer Society was founded. It was first the American Society for the Control and Prevention of Cancer, I believe, and it was founded as a group, basically as a private army. You see here in this very early American Cancer Society poster, the Women's Field Army. It was clinicians, mostly surgeons, working with women who really went door to door and said, we need $1 to be a member of this society that is going to make sure that cancer is cured. 1914. In those years, 1920s, cancer really, you had 2 hopes if you had a solid tumor. If you had a liquid tumor, it was done. Leukemia was completely fatal. If you had a solid tumor, you had 2 hopes. Either a surgeon could cut it out, or radiation therapy, which was pretty brutal at the time. This is one of the 1920 radiation delivery devices. Pretty brutal, like at the time. Those were your only hope. And if you think about what was going on, this is just a snapshot from 1937, a snapshot from what happened over the decades. Here we are up until about 1937, where that was the year where the diseases due to infectious disease, deaths due to infectious disease fell below deaths due to cancer. 1937. So before 1937, more people died of infectious disease than died of cancer in our country. And you see here the little blip of the 1918 influenza epidemic. And in 1937, the first National Cancer Act was passed. And it created the National Cancer Institute. For the first time, the federal government getting involved in funding cancer research. Before, it was societies, the American Cancer Society, the AACR, the American College of Surgeons, which had a very prominent role in funding cancer programs. But in 1937, the government got involved, formed the National Cancer Institute. And then around the time of World War II, something very dramatic happened, and surgeons have a role here too. There was nitrogen gas left over after World War I, was in the U.S. fleet, in the hold of the ships that were docked at the U.S. fleet in Bari, Italy. The Germans bombed the fleet. All the nitrogen gas dispersed into the town of Bari, Italy. And the citizens of Bari, Italy died terribly weeks later. And two pathologists, a group from the US Army, came over to do autopsies on the citizens of Bari who died. And they found that they had a complete lack of lymphoid tissue, in addition to the toxic effects, respiratory effects of the gas. The ones who died slowly had no lymphoid tissue. So two pharmacologists, Alfred Gilman and Louis Goodman, started working on this, developed a derivative of nitrogen mustard called L-mustine, and used it to propose a new treatment for lymphoma. What happened next was a surgeon, a thoracic surgeon, Gustav Lindskog, who was working at Yale University. Al Gilman was there at the time. And he took whatever dose that Gilman helped him calculate a dose from what he'd been giving to his mice, and gave it to a patient who was dying from a thoracic lymphoma. And these are from his actual notes that come out of the Yale archives. On August 27th, 1946, at 10 a.m., the first of 10 daily doses of chemotherapy was given. This patient was in severe respiratory distress from his lymphoma. 4 days later, the patient said he could sleep, eat, and move better. By September 6th, he had greatly improved. With blood transfusions to treat falling blood cell counts, he continued to improve. On September 27th, all cervical and axillary nodes of the cancer were gone. Dramatic response to L-mustine in this patient with non-Hodgkin's lymphoma. The patient went on to recur. L-mustine is not the optimal drug for treatment of Hodgkin's disease, of non-Hodgkin's, thank God. But this was the first chemotherapy that was ever given to a patient that ever worked, was given by a surgeon, and I think we should be very proud of that. The early years were about surgeons demanding that you follow case histories very carefully so that you can make sure that your treatments are working. In 1944, something dramatic happened because there was a recognition that that is not good enough. We believe in our treatments, and so we can be biased. And so in 1944, the first randomized control trial was conducted. It was done by this gentleman who's a statistician, actually the first statistician in clinical trials, Austin Bradford Hill, a Brit, did a trial of streptomycin versus bed rest for tuberculosis. And the people on this side of the pond were paying attention to this, thinking, we have new chemotherapy drugs, we have new agents, we don't know if they're going to work, and they tend to be very toxic and hurt people. Maybe we should use this technique so that we can be on solid moral ground for putting people through this, because we're going to test in a blinded, very objective fashion whether we're helping them or not. And that was incredibly profound at the time. And I think the only reason we could go forward with moral authority with the kind of things that happened during the early years of the development of chemotherapy. In 1948, about that same time, Dr. Hamblin joined the American Cancer Society Georgia Division, became very interested in work that the Cancer Society was doing in its intramural program in cancer epidemiology. Many large studies were being run, the cancer prevention studies that ran for many decades, including studies that looked at the issue of smoking and its association with lung cancer, nutrition in cancer, and throughout the decades, these have continued to provide wonderful, huge, important cohort results that have informed our health policies. 1948 again, just as chemotherapy was hitting our world and in its infancy, Mary Lasker, a wealthy Manhattan socialite, noted the NCI budget, $500,000 in 1948. And this is a wonderful quote from Mary Lasker. Just, $500,000 was a lot of money in 1948, but here's what she had to say about it from her perspective as the wife of a very famous businessman. In business, $500,000 wouldn't even be a suitable sum to use for an advertising campaign for a toothpaste. I was appalled that no single laboratory had this amount with which to try to conquer the number 2 cause of death of the people of the United States. It was only a decade before that had turned into the truth instead of infectious disease. So Mary Lasker launched a campaign with Congress, went straight to Congress, and was able in 1954, not that much later, to achieve congressional funding of the National Chemotherapy Program. Again, role for surgeons in this progress. You see Isidor Ravden next to General Eisenhower as his surgeon. And when Eisenhower needed someone to run the new cancer chemotherapy program, he turned to Dr. Ravden, and Dr. Ravden founded at the NIH he became the chair of the clinical studies panel of the National Chemotherapy, the Cancer Chemotherapy National Service Center. And the first thing he did was at the same time, another very important observation. The first half of the 20th century, we as surgeons were surgery and radiation were the only tools we had, and we used them as fiercely as we possibly could. Bigger surgery, more surgery, could hopefully save more people. Well, many people started to realize that this just wasn't the way that cancer worked, wasn't the biology we were dealing with. 2 brothers, a surgeon, Bernie Fisher, his pathologist, brother Edwin, came up with an alternative hypothesis in the 50s that tumor cells don't creep along like crabgrass. Tumor cells jump, they embolize and go throughout the body, and cancer really is a systemic disease, perhaps from the very first time it becomes invasive. And so really bigger surgery and radiation is not going to do what we need. We as physicians treating cancer need to try to do something else. So Dr. Ravden's team, working in chemotherapy at the NIH, together with the ideas of the Fisher brothers, founded something called the Surgical Adjuvant Chemotherapy Breast Project, rounded up their friends, surgeons at 23 institutions, and got together a team to try to do something about breast cancer. And the very first breast cancer adjuvant trial was conducted from 1958 to 1961. The 3 main participants were Bernie Fisher, Nelson Slack, and Norman Wollmark, who to this day is still one of the leaders of the NSABP Foundation. And the study it wasn't a great chemotherapy, but it was pretty good for the time, Thiotepa versus placebo, as an adjuvant to, look at this, radical mastectomy in 1957, still the days of very, very big surgery, in the treatment of patients with primary carcinoma of the breast. So then in 1971, shortly after that trial was launched, Nixon announced the war on cancer. And you see here at the top Nixon signing the very famous picture of Nixon, but also important, you see Dr. Hamblin, who was the president of the American Cancer Society at the time of the Nixon signing of the 71 War on Cancer document. And Dr. Hamblin is the one that Nixon turned to after he put down the pen to say, okay, now, boys, go to work. And you can see what Nixon said here. I will ask for an appropriation of an extra $100 million, that was a big sum in 71, to launch an intensive campaign to find a cure for cancer. The time has come in America when the same kind of concentrated effort that split the atom and took man to the moon should be turned toward conquering this dread disease. We've heard a lot of criticism over the years of Nixon's war on cancer. Hubris, thinking this was going to be easy, thinking, it's putting a man on the moon is hard, kicking cancer can't be that hard. But I am a passionate defender of the good that public funding has brought to the world of cancer treatment and research. And there are many things that have come out of the war on cancer that we can say have led to the progress that we have today. It funded basic science research. It expanded comprehensive cancer centers. It established multi-institutional cancer clinical trials networks, such as the ones that many of us work in today. It supported nationwide cancer control initiatives, such as the ones that Dr. Hamblin was so instrumental in leading. And it led to a decline in cancer mortality. Not a cure, but a absolute decline in mortality. And I'll show you a little bit about the effects of that in a bit. So what about the very first adjuvant clinical trial conducted by that group of surgeons where, well, here's the result. This is the 10-year follow-up result. It's Thiotepa, pretty ineffective chemotherapy, but better than nothing. 21% better survival for the patients who receive Thiotepa. Underscore here, inadequacy of standard operative therapy in affecting a permanent tumor-free disease state in the majority of patients. Radical mastectomy, this group went on, obviously, you all know the history, to eliminate radical mastectomy from our repertoire and actually prove that with chemotherapy you could do less and less surgery with equivalent, or maybe even when it comes to side effects, superior efficacy. And then finally, this very sobering observation: cancer is a systemic disease. 76% of participants with positive axillary nodes demonstrated a treatment failure by 10 years, and only a quarter of these survived. So we had to do better. The American Cancer Society was very busy during these same years too, doing cancer control work. Here's Dr. Letton's work again. 20-year follow-up of the breast cancers diagnosed during the Breast Cancer Detection Demonstration Project. This was a project, like many of the things that the American Cancer Society did, that kind of already had the idea that they knew what the right thing to do was. So they weren't randomized clinical trials. They were prospective cohort studies asking people to do something that they thought would be positive. And this one turned out to be correct. This is just a study. The blue line are women who participated in this demonstration project, which basically was go in, get a mammogram, and act upon the results of the mammogram with your physician. And that showed a superior survival compared to just general SEER data. And so the results of this showed that it accomplished its purposes. It placed breast cancer screening in the consciousness of providers and women throughout the United States. The rate of advanced cancers decreased considerably, allowing greater chance for cure, and it decreased breast cancer mortality by 6.3%. We still don't know for sure. As you know, there's been a lot of controversy over mammographic screening and what it really does in 2018, but you can say unequivocally that in the era when it was absent compared to what we see today, it definitely contributed to a reduction in mortality for this disease. Okay, so this is about 1971 when the results came out of that very first NSABP trial. Thiotepa is better than nothing in adjuvant treatment of breast cancer. And then if you take into account about a 10-year lag of adoption and being used and being able to track it, you see the first arrival of chemotherapy for women with breast cancer and the widespread arrival of mammography, and then you start to see a decline in death due to breast cancer. And we've seen this in many of the initiatives of the American Cancer Society and that came out of the war on cancer: smoking cessation, mammography, colonoscopic screening, and adjuvant chemotherapy. And so solid tumor mortality is falling, and it has fallen as a result of these changes, which happened decades ago and then have matured over time. So what's happening a little bit about where we are with cancer clinical trials and clinical research today? Well, back in the 1900s, Halsted era, cancer research was the team conducting the research. It was an individual hospital, an individual institution. The team treating cancer, the surgeon, the radiation therapist, and the pathologist. The studies were case series with careful follow-up. Then in the mid part of the century, randomized controlled trials came, the advent of the biostatistician, and prospective longitudinal cohort studies such as the ones that Dr. Lenton conducted with the American Cancer Society. And the team treating cancer and doing cancer research changed. Now there were the multi-institutional consortia such as the NSABP, and the surgeon, radiation therapist, and pathologist were joined by this new field, medical oncology, and by the statisticians. Well, where are we today? Today, things have gotten a whole lot more complicated in the last decade. We now have clinical trials designs that are wide and varied and numerous. Marker-driven adaptive designs, comparative effectiveness research, which allows us to do research that we've been unable to do before just because there are certain conditions for many reasons cannot be randomized, surgery being one of them often. And we now increasingly are doing studies for molecularly defined disease cohorts. Really challenging. Every tumor we treat now is becoming a rare tumor in trying to gather data that will allow us to know what to do. And the multi-institutional research consortia now spread across the entire spectrum of care. Half of the institutions participating in Alliance clinical trials are community oncology institutions. So it's no longer academia doing all this work. We are partners, very careful partners with the community surgeons and community oncologists. And the team is now huge and varied. Basic scientists are front and center into research these days, bioinformaticians, imaging researchers, molecular geneticists. Patients are now actively involved in designing and conducting research. That's been a recent and really interesting entry into the clinical trials world. Information systems experts. If Amazon knows everything about your spending habits, we need to know everything about how doctors are treating cancer patients. Harnessing that kind of knowledge to figure out what to do next is a next step we need to take. So what's happened? This doesn't come without a cost. That $100 million that Nixon put in for the war on cancer back in 1971 would not even pay for a single pharma-run adjuvant breast cancer trial. Not one single trial can be paid for by $100 million. In 1948, the funding came from the government, usually the NIH and the state. Private academic institutions put their own skin in the game to conduct this kind of research, and private foundations such as the American Cancer Society were major contributors. In 2018, there has been government expansion into research. That occurred over the last half of the 20th century. Academic institutions and private foundations are still very important. But there are now some really big players in the space of cancer research, industry, and venture capital. And I'll show you a little bit about what's happened. So I cut off the good part of this graph, the huge every decade doubling of the NIH budget that occurred in the latter half of the 20th century. And then about 2003, the National Cancer Institute hit flat funding. And the funding levels you see at the top, meaning flat funding, and the effective funding levels in what equivalent dollars would have been back in 2002 have been falling. So funding flat, which means, but we can do less for the dollar. So government funding, at least for the National Cancer Institute, has been falling since about 2003. What's been happening in industry? Oh, and by the way, you look at this total funding level of the NCI, $5 billion. Now we look at the yearly funding of pharmaceutical companies and their research and development spending. And here is 2018, $155 billion are spent by the pharmaceutical industry in that year, in this year alone. And the percentage of that funding that is going toward cancer is about a quarter. So the pharmaceutical industry vastly outspends what federal funding does for cancer research. And as no surprise, there's been a huge proliferation of drugs. This is great. This is wonderful. Look at this. This is the tracking from the FDA of FDA drug approvals. Look at this exponential growth we've seen since 1995, and these drugs are now highly varied. The dark black shows cytotoxic drugs, but now you see these lighter colored ones in the middle are the biologics, and these are the targeted therapeutics. So we're seeing now 50-50 development of cytotoxic versus biologics. And 2017 saw a record number of FDA approvals, with cancer leading the way, 16 primary oncology drugs and 11 hematology drugs in a single year alone. So it's no surprise with that much capital being spent on developing new treatments, we're seeing a major expansion in drug therapy for cancer. Well, like I said, this is good news. This is immuno-oncology, just the new indications from 2011. In the field of immuno-oncology. We, in the cancer clinical trials world, still don't know what the collective impact is going to be of the entrance of immuno-oncology into our armamentarium, but all the early indications are it is going to be truly dramatic. I mean, I think we all know of a very famous citizen right now, Jimmy Carter, who's dear to many of us, who had a very advanced stage melanoma, who we all believe is doing pretty well today because of the arrival of immunotherapy for that disease. Never would have happened even 5-7 years ago. We also have some new diseases as a result of these new targeted therapies, brand new diseases. Mismatch repair cancer. MSI high is now a new kind of cancer. It spans different disease organ of origin, and there is a single FDA-approved treatment for mismatch repair high cancers, the checkpoint inhibitor pembrolizumab. There's another one that's on the horizon, and I think we're going to see more and more of these as particular molecularly targeted drugs achieve efficacy across different conventional disease states. This one is the TRAC2 inhibitor trial. There are a whole bunch of them ongoing. Early results show that in joining microsatellite high disease, we're going to have TRAC2 fusion disease, or they'll come up with some probably fancier name for it. So we're going to have a whole lot of new diseases, and we're going to have to figure out what role surgery plays in managing and treating those new diseases, because I'm sure you all know, every time medical therapy changes, surgery doesn't go away. And in fact, in many, many cases, the more effective medical therapy is in solid tumors, the more surgery we end up doing. Because we end up now in an era of surgeons being able to manage metastatic disease that is progressing, because the overall systemic spread of the disease is generally controlled by drugs. Really fascinating, and it's going to be a big challenge for us to know exactly how we handle the challenge of understanding when's the best time to do surgery in this new era. So fortunately, we have a lot of tools to start to study these issues. Clinical genotyping is now wide. I'm sure all your patients with cancer come to you saying, here's my foundation, when's my foundation medicine panel coming back, and what are you going to do about it? We have great value in our clinical outcomes databases. The Commission on Cancer of the College National Cancer Database is getting more and more valuable by the day as we need these data. Many new clinical trials designs that can address very different issues. It's not just simple randomized treatment A versus treatment B, because this is a much more complicated situation we're dealing with. Functional imaging, give a drug, take a picture, is the drug working or not? Much better way than waiting for the thing to shrink. Cell-based therapies, that's been the big news of this year, and we're seeing just a revolution in the way some cancers are treated. Very optimistic initial results. No idea where this is going to land eventually, but we'll know in 4 or 5 years. And then finally, the electronic health record. Patients reporting their own outcomes. There's so much that can be done with the new information technology that I think was just a hope when we first thought about the National Cancer Database, and now we might be just getting it through the EHR in a much more straightforward manner, hopefully. So what do we have in 2018? Our field, our work, is now migrating towards centers of excellence. We have technology development in innovation labs, new procedures, new surgical techniques being developed in innovation labs. The clinical trials research groups are incredibly busy, as you might imagine, with this wealth of new drugs. Cancer centers, the National Clinical Trials Network groups, outcomes research and comparative effectiveness research. The American College of Surgeons has many prominent programs for those, as does the University Medical Centers, the hub of development of that kind of work. Translational research remains incredibly important. We wouldn't have new categories of disease based on molecular typing if we didn't start seeing success from translational research. And then finally, there's a whole, well, how are we going to pay for this? How are we going to do this? How are we going to get cell therapies to children with leukemia who need it? So now, entered into into cancer research are now business schools of public health, to figure out how to use our tools, these very expensive tools we've developed, and use them well. So we have a lot to be proud of as surgeons. 100 years of surgeon-led translational cancer research. I'm just going to flip you through some of the highlights. Surgeons set the scientific foundation for tumor immunology, which is now reaping incredible benefits in our new immuno-oncology world. Metabolic support of cancer patients. Being able to treat patients through the cancer treatment process was a transformative advancement that sometimes we forget. Now that it's so easy to see where we are today, we forget how impossible any of this would have been without this progress. Surgeons also first uncovered the associations between toxins and tumor formation, and much of public health and the legacy of Dr. Letton's work in the American Cancer Society continues in focusing on epidemiology and prevention of cancer by prevention of the genotoxic insults. Hormonal modulation of cancer, for which a surgeon won the Nobel Prize, adjuvant chemotherapy for solid tumors, all championed and accomplished by surgeons, stromal involvement in metastatic disease, and finally, refinement. Never to forget, refinement of surgical technique to reduce treatment morbidity. We're getting a lot less brutal, hopefully, in what we have to put our patients through, and we have a lot of reason to believe that those kind of advances will continue. There's some things that I think it's wonderful for surgeons to continue to think about, although I would never want to put us in a box and say this is our space, but we are surgeons after all, and we have a particular approach to the field of cancer that I think is very valuable and we should own. Response to neoadjuvant therapies. I'll describe a little bit more about that in a second. Define the clinical behavior of molecular subtypes. I've been working with this group at the Dana-Farber for a long time that does lymphoma research, and they laugh at me. I'm the lymphoma surgeon. Because they give these drugs and 5 of the lesions are melting away and 1 is growing like crazy. So they turn to the surgeon and say, get that one so that we can figure out why that one is growing and the other 2 are shrinking, and the others are shrinking. And we're doing that kind of work all the time. And guess what? If the team doesn't have a surgeon really committed to answering those, it's kind of mundane. You could send them to the interventional radiology lab and have them just, spear those tumors. But the difference is, at the same time, we're removing that disease for many of the solid tumors, and then looking at the long-term effect of what happens to that patient down the road. So I don't want to launch into too much length here, but this is what I see as one of the great frontiers for surgical oncology in the at least near future. Gastrointestinal stromal tumors. There'll be 10 lesions. 8 of them are shrinking, 2 of them are growing. You go take out those growing ones and let the chemotherapy take care of the rest of them. Is that going to make a change to that patient's overall outcome? And the truth is, we don't know the answer yet. We think we might know, we don't know. And so who's going to study that? It's not going to be anybody but us, and it's not so easy to do. So a lot of what we need to do is going to be very challenging in terms of design, in terms of collective commitment to answering the question. That's just one example. Management of post-chemo residual disease, I think I already talked about that. Characterization of treatment resistance. These are the key questions in oncology right now, and surgeons can play a tremendous role. Surgery versus observation for borderline neoplasia. We know we are over-treating prostate cancer, we're over-treating breast cancer. How many other things are we over-treating? Who's going to figure out what we're over-treating? It's going to fall to the surgical community. Tissue engineering for functional reconstruction. This obviously, I haven't had time to go into that, but a great advance from our community. And the relative benefit of new techniques, devices, and modalities. We are getting a lot better at taking very careful looks at what kind of outcomes are we getting out of the robots. How do we rationally develop and promote new technologies, especially if they're expensive? So for the more junior people in the audience, what do you need to do to have a skill set that you can take forward to do cancer clinical and translational research? Well, you need to be a cancer surgeon. You need to know everything that surgery can teach you and has to offer. You need to understand a bit about tumor biology and how that relates to what you are doing as a surgeon. Clinical trials methodology. It's a really fascinating, interesting subject, and there are some very different approaches that we need to take to answer the questions that surgeons want to answer that are different than approaches than what the medical oncology community adopts. Molecular pathology, imaging biomarkers, drug and device development, pretty complex space to be in, laboratory medicine, regulatory affairs, and very important, understanding funding opportunities. It's so sad to see people with really brilliant, wonderful ideas that can make a great difference who just don't have a way to get it funded and supported. And that is something that I think the groups such as ours can help with to a certain degree. Although, just one last editorial pitch here, that pharmaceutical budget that's going up like crazy is not helping us with the kind of questions we need to address as surgeons. The federal budget that is remaining very flat is what supports the kind of research that we need to do in the comparative effectiveness and in the treatment development space that don't yield pharmaceutical companies big payoffs when they have a new drug to market. So we all need to continue to advocate very strongly for federal support of our research, because that's where research that surgeons by and large are involved with comes from. So finally, 2 examples I'm going to show you. I think both of these are transformative for the field, and both of them were conducted by surgeons. This was an alliance trial, randomized phase 3, the latest iteration of the legacy of Norm Womark, Bernie Fisher, Isidore Ravnin, is this 2,300 women with a tissue biopsy showing an early breast cancer. All of them had their tumors tested. 460 had a HER2 positive tumor. So they got randomized to either cytotoxic chemotherapy plus an antibody targeting the HER2 receptor, or chemotherapy plus the antibody plus a small molecule inhibitor, a double hit for the HER2 receptor. And then they went to surgery. And this didn't get a lot of fanfare at the big cancer medical oncology meetings, but was an amazing game changer for all of us. The primary endpoint of this clinical trial was the presence of tumor in tissue removed at surgery. This was a study of neoadjuvant treatment. And what really transformed this was then the FDA looked at this, combined it with many other adjuvant studies, and put out a document, a guidance document in 2012, saying that pathologic complete response in neoadjuvant treatment of high-risk early-stage breast cancer is an endpoint that can be used to support accelerated drug approval. So if you could make this tumor go away with chemotherapy, and surgeons could show that, that was an endpoint in drug development. And we've seen an explosion. But think about this from a tumor biologist, from someone who wants to understand the biology and how it works. This is the way to understand tumor biology, and it's in our hands. You treat the tumor and then see what happens. You don't take the tumor out, treat the patient, and God forbid, hope it never comes back. And so that's the shift that we've seen in many of the solid tumors that's making a tremendous difference. First of all, in our role as surgeons, and second of all, in our ability to really understand what the biology is. I want to see this happen for colon cancer. I have yet to convince my colleagues that we need a neoadjuvant protocol for colon cancer and do for colon cancer what breast cancer has done. There's a lot of resistance out there. Guess what? It's only the surgeons who are going to make this happen, not our medical oncology colleagues. So I'm putting a challenge out there to you colon surgeons. Here's another one, you know, the surgery branch and the intramural program at the NCI. Steve Rosenberg has been laboring away for so many decades in the belief that the immune system can conquer cancer. And this is the surgery branch. And I am so proud of what his group has finally been able to achieve. This is just a study that came out 2 years ago. 101 patients with metastatic melanoma, including 76 with metastatic disease. You can't even randomize to an arm with no TILs now. They work so well. There was a non-myeloablative chemo plus TIL arm, and a non-myeloablative chemo plus some total body irradiation plus tumor-inflating lymphocytes. And this was presented by a former fellow in our program who now works with Steve 2 years ago. The complete response rates, remember 76 of these had metastatic disease, were 24% in both rates, complete response. Overall survival was similar. With a median potential follow-up of 40.9 months, only 1 of 24 patients who achieved a CR recurred at all. How wonderful that we're finally seeing Steve's program producing these kinds of results. Let me just end quickly. I'm a little bit over time. I remember I promised to show you that Dr. Letton's legacy, the work that was done in the latter half of the 20th century when we didn't have as many tools, when we had cancer control programs and simple, relatively simple things like mammography, it worked. It paid off. Look at this. Mortalities are falling. This is not because of all the fancy stuff we have today. It's because of their hard, long work. And if you look there, this is just an analysis that was done by the Cancer Statistics Group at the American Cancer Society, showing that if you just showed how that curve got bended, how many lives were saved, and this is up to 2014, 1.5 million men and over 660,000 women had their lives saved because of the results of individuals like Dr. Letton. So let me just end with one more challenge. This is the government again, and this was the 2016 State of the Union address by President Obama. Tonight I'm announcing a new national effort to get it done. Because he's gone to the mat for all of us on so many issues over the past 40 years, I'm putting Joe in charge of mission control. For the loved ones we've all lost, for the family we can still save, let's make America the country that cures cancer once and for all. And then when Joe came to ASCO as a guest last year, he did it really well, because this is what I think it's going to take in his humble fashion. Imagine if you all worked together. I'm not kidding. So thank you very much. Such a wonderful honor to be here. I really appreciate it.